breaking down CIBIO with macroecology and biogeography of fishes.
Thanks, Marta. Thanks, everybody. Well, I have to talk a little fast because Renata talks a lot. But, okay. Well, I'm from Brazil. I spend my life there. Now I'm in Perpignan, in France, doing my PhD. I worked with the ecology of fish, reef fish in general, especially herbivorous reef fish. Now I'm increased the scale a little bit. I'm working on macroecology on a more global scale there in Perpignan. So I'm presenting here some of the results, some of the things I've been doing the last this year. Well, yeah. First thing is what we have to define what's a herbivorous fish. There are many. It's not easy as we may think at first. So in terrestrial system, it's quite easy to see what's an herbivore. They are those animals that eat plants or plant-like organisms, especially primary producers. But in the sea, it's not that easy. It's not that simple. But especially in reef environments, the fish are the most important herbivores. Why? Okay, so there's a great variety of types of herbivores. They are present in different families, and they have very different diets. So there are those species that ingest primarily algae, so they're the true herbivores in some sense. But in reef environments, most of the reef is covered by a very tiny, a very thin layer of what we call algal matrix, which is, okay, it's not necessarily only algae. There's filamentous algae, but the detritus, invertebrates, bacteria, and all types of things that most of the herbivores are eating this type of thing, this type of food. So that's why it's not. I mean, these fishes, they do not eat. Most of them they do not eat primarily algae, but they are eating, in fact, these detritus and bacteria and all types of things together. And there are of course some fishes that are omnivores, but they eat a lot of algae, and most of the time we consider them as herbivores as well. So okay, so for the sake of this talk and for the things that I've been doing, we consider herbivore as every fish that ingests any type of primary producers, algae, seagrass, cyanobacteria, and so on. It's a very broad concept, broad classification. So why is it important to study this fish? Herbivore is not only on coral reefs, but in subtropical and even in temperate reefs. They are among the most important trophic group in terms of abundance and biomass. They compose a huge proportion of the structure of these assemblages, and they have a very important influence in controlling the distribution and the benthic community in general. So by eating the algae or the algal matrix and so on, these fish, they can control, in some sense, they modulate the benthic structure of the places that they live. Same time, not only herbivores, but every time they're being more fished, as many other groups. So we don't know exactly what may happen to a system when we remove these fish. They're being removed every time more, and we kind of we must know what is the consequence for the system, for the reef, if we are keeping removing them. Okay. So they're primarily very important. I wouldn't say key species, but they're very important in structuring the community in general. Okay. As it may, well, despite their great importance, we don't know how they are distributed on a very global scale, on a very large scale. We know more or less how are the herbivorous assemblages here and there, but we don't know what are the major macroecological patterns, and we don't know what drives these patterns. So based on that, my aim was to describe these patterns on a very large scale, on a global basis, and especially some of the traits, some of the most, what we think are the most important traits for this fish, and to correlate these patterns with a number of factors, environmental factors. So, okay, I used a database which was already compiled. I was not part of this group, but it was a Gaspar project which was led by some people from France, but involved the people from Brazil, people from Australia, and different places in the world. So their main aim was to compile information from, at first, 169 locations worldwide, and these are the dots here. So for each one of these locations, we have a very precise, a very good list of fish that occurs in each of these places here. At present, I've been expanding this list, and now we have approximately 270 locations. But the things I've been showing, that I'll show here, is only with this database. Okay, from this database, the sites in here, they were classified into 6 biogeographical regions based on the similarities and differences in the fish assemblage. So we have here the Central Pacific, the Eastern Pacific, the Western Atlantic in general, including the islands here, Eastern Atlantic, Western Indian, and Central Indo-Pacific. Okay, this is a very important thing that I'll go back later, these regions. And in the world, okay, including, not in the world, but in this zone here, in the mainly tropical zone, there are about 6,300 species of fish. So from this amount, from these 6,000 and so on, we classified 550 as herbivores using that very broad concept. And they are distributed within 16 families. It's a quite, relatively good number of families. Although most of them are restricted to 5 or 6 families only. First thing we classify them according to the 3 feeding modes, feeding behaviors, which we call. So these 550 species were classified either as browsers, grazers, or scrapers. Unfortunately, we don't have the diet for all the species, so we couldn't create a classification based primarily on diet. So we classify these according to the way that fish feed, which is in some sense related to diet, but not necessarily so. So here the browsers are the fish that eat basically macroalgae. So in some sense they would be the true herbivores. Most of the fishes here, they have endosymbionts, bacteria that breaks down carbohydrates and so on. That's their primary way of feeding. And these grazers and scrapers, they are, in some sense, they have more mixed diets. They eat mainly in the algal matrix I mentioned. The difference being that the scrapers, they are for being larger, they usually scrape the substrate. They create marks. They create a higher impact in the they remove more material than the grazers here. So it has there's important implications for the dynamics of the environment, different way they feed. Okay. So, okay, for each one of those 100 and something sites, we calculated the number of herbivores, the proportion of herbivores related to the total richness of the site, and the proportion of each one of these 3 feeding groups in relation to the number of herbivores, of course. So we had more or less this, how is the structure of the herbivorous assemblages in each one of these 169 sites. So each one of these variables here are correlated with the biogeographical region. We had the sea surface temperature, the level of isolation, the area of coral reef and the coastline for each one of these 169 sites. We use different types of models. I'm not going to explain the type of model and what we did, but we modeled each one of these things here in relation to these variables here with different approaches. So this map here, we have the 169 sites, and here shows the richness of herbivores, the number of herbivores in each one of these sites. There's a very clear pattern here in the sense that these regions here, the Eastern Pacific and the Atlantic in general, there are fewer species of herbivores. You see that at best around 25 to 50 species here, while in the Western Indian and the Pacific in general, the values are larger. So there are more herbivores here than here. It makes some sense because here the richness, the local richness is much higher here than here. And when we see in the proportional terms, so it's a way of kind of controlling for the local richness, the pattern is more or less the same. There are some exceptions here. You see the proportion of herbivores is much smaller here. But there are some exceptions, some sites with a higher proportion of herbivores. Here the same, the proportion is in general very larger, but with some exceptions here and there. So I'm not going to show you a table with the AXE values and nothing like that, but the thing is that the message is that for the number of herbivores and for the proportion of herbivores, the biogeographical region and the coral reef area were the most important factors for explaining this. So if we want to know how many herbivores and what's the proportion of herbivores in a given site, if we know where it comes from and in the coral reef area, we can have a very good idea. After that, remember that I mentioned the 3 different feeding groups. I use the same approach for these 3 groups and for the browsers too. So this is the proportion of browsers per site, and here is the relative influence of each one of these factors, explaining this pattern here. Interestingly, the sites, the areas where the number of herbivores is low, in the Eastern Pacific and the Atlantic in general, the proportion of browsers is higher. And here in the Indo-Pacific in general, the proportion of browsers is higher in peripheral areas. When modeling these, we see that the most important factor here wasn't region, as we would expect, but instead it was temperature. But there was a negative relationship between the proportion of browsers and temperature. It means that the colder the water, the more browsers there are in one given site. And remember that these are the true herbivores, in the sense that these guys are the ones that eat macroalgae. Okay, for the grazers, the pattern was the same for the number and proportion of herbivores. The higher proportion is here in the Pacific in general, and region was by far the most important correlate. For proportion of scrapers, there was an interesting pattern because the proportion of scraper was much higher here in the Atlantic than in the Pacific. Region wasn't again the most important factor, but in proportional terms, the assemblages in the Atlantic are dominated by scrapers and browsers, while in the Pacific they're dominated by grazers. So there are disparate structures in the herbivorous assemblage depending on the region, and of course there are different factors regulating this. Okay, this is just to show how complex things can be. Those models, they were very, they were simple, more simple. Anyway, here it shows it's a structural equation model. It shows how everything is how things interact with each other. And interestingly here, although temperature was not important for most of the factors, there is a very important indirect factor in the sense that temperature can be correlated to calorific area, which is correlated to coastline, and that is correlated to the herbivore richness and so on. I'm not going to explain everything, but just to see that some indirect factors, some indirect correlations occurs here. It's important to take that into account sometimes. Okay. Apart from the feeding mode or feeding behavior, another very important trait is the size of the fish. Not only fish, but any species. It's very important to know how the size of the species is distributed. Okay, based on that, the question here was to know how what's the structure of body sizes in this fish, in this group. Here I have a very basic histogram of the body size, and this in white. The body size of all the fish, all reef-associated fish. It has, of course, a normal distribution, and here in green is the body size distribution of only the herbivores. It's interesting that to know that there is no herbivore very small and or very large, so they're more or less in the center of the distribution. But instead of having a normal distribution, there's a kind of bimodal distribution here. Okay, now I split this distribution. So here in gray is the distribution of herbivores, and I split the body size of the browsers, the scrapers, grazers, and the scrapers. And we can see that the browsers, those that are more correlated to cold waters, and those are the true herbivores. They tend to be larger. The grazers, those that dominate the assemblages in the Pacific, they tend to be smaller, and they are responsible for this mode here. And the scrapers, which are more important in the Atlantic, as you remember, they are larger as well. So one thing that we want to know, and I'm not going to show you results because we still don't have these results, is what drives this difference. Is there any why this structure here is different? Is it related to the phylogeny of the species, the way they feed, what they feed, where they live? We still don't know, but this is something that we are looking for. Okay, so is there any influence on diet? For instance, I told you that these guys are the ones that eat the algae, and they ferment these algae. They need to eat a lot because the pattern for mammalian herbivores is the same. For instance, mammalians, which are herbivores, they tend to be larger because they need to eat a lot of food for digestion, for processing it. It seems that among fish it's the same. It's a possibility, we still don't know. While these guys here, they are eating more or less the same thing, but they have very different size patterns. So probably there's a phylogenetic component here that are driving this difference. I mean, here are just more questions than answers in this part here. Things that we still don't know, what's the importance of diet or the phylogeny, and one thing that we are looking for as well is to compare the size structure of tropical against temperate regions. I told you that we are putting more sites on that first database. We're putting more special temperate sites, so in the near future we'll have a better picture of how is the size structure of tropical against temperate areas. Okay, well, I showed some things, some results and so on for two traits. One is the feeding mode or feeding group. The other one was this for the body size. We are looking at the functional structure in these assemblies, and for that we are using an approach which we're not directly interested in the species, but on the functional entity or the functional group that is a species corresponds. So hypothetically, just to explain you a little bit, I don't know how we were or not on this thing, but let's suppose we have here 5 different species, A, B, C, and D, and I can put here 3 different traits, trait 1, 2, 3, not giving names, but just hypothetically. Let's say that the first trait is a square or a rounded square here. Species A is a square, and B and C and so on. So the second trait is to be 5 or 10, let's say, star. Yeah, tip stars. So, and the third trait is to be a green, yellow, or let's say or blue circle. Okay, so each species has one of these traits. A trait is something that we can measure. We can measure preferentially on an individual level, but we can be measured in a specific level as well. So by combining these traits, we have what we call a functional entity. So instead of using species A, we are using this functional entity here. It's a square, a 5 tip stars, and a green dot, a green circle. And with that, we can put together, combine the species based on their functions, which means that the species B and C here, they have the same combination of traits, so they belong to the same functional group or the same functional entity. This is very important because we're not, in some sense, because I mean, if these traits they are representative of what the species do in the environment, they are basically the same. It means that if I remove the species C, this function here is already okay. It's still going fine because we still have species B. Get it? So we can start measuring how redundant or vulnerable a community is. So imagine one community or one assemblage where each functional entity is composed of only one species. It's a very vulnerable community or assemblage in the sense that if we remove one functional entity, if we remove one species, we are losing one function. On the other hand, if we have an assemblage where we have lots of species within each functional entity, it's very redundant in the sense that if we remove one species, we're not losing that function. Okay. Another thing is that we can start to see how the functions are related to each other based on their distance. For instance, this function here and this here, they're almost the same. They have only one trait which is not the same, which is different. While we have this function here and this here, they are totally different. So how do we measure if two entities are close or far? That's another thing. We can measure how variable these functions are and so on. So for that, I went back to that database with 550 species of herbivores. For each species, we assign six traits. So the feeding behavior with 3 levels, those browsers, grazers, and scrapers that I mentioned. We have maximum body size of the species with six different levels. We have schooling behavior. Species is solitary, lives in pairs, in small groups, and so on. The home range of the species, if it's very site attached, it has a medium home range or a very large one. The period of activity, whether it's diurnal or nocturnal species, and the level in the water column, if it lives near the bottom or high in the water. 3 levels. So by combining all these levels here of these 6 traits, we have 1,260 potential functional entities. It means that we could have this amount of functional entities, but we only have 87 represented within the herbivores. Okay, but after that, what we do, we can, using more or less the same approach of analyzing the community with the species and so on, we can use these traits. Okay, we can build multidimensional space with a multivariate technique and so on, and we can calculate the volume of this function. I'll show you this. Okay, this is a plot with this is the principal coordinate analysis, well, the 2 axes for the principal coordinate analysis for all the herbivores in the world. Doesn't mean much if you look like this, but once we have this, we can start to map the species. So where the species is and how close they are in this plot here. For instance, these species is far from these ones, and so on. More interestingly is to see how, okay, this is the structure for the whole world, how this structure changes according to the region where they are. So, okay, and we can calculate the volume, we can calculate the convex hull that these, but this is a 4-dimensional analysis, so only showing you the first 2 axes, so it's not very complex thing. But we can calculate the volume of that these functional entities they compose. Okay, remember those 6 regions here? Here is the plot with the outer line here is the functional volume of all the herbivores in the world, and in red here is the mark. There are only the species which are here in the Central Pacific, this region here. Okay. And we can, of course, this is the axis 1 and 2 of the analysis, this is the axis 3 and 4, so we can have an idea of how is the feeling of that hypervolume that we have, how is the feeling of this volume in each one of the regions. And we have, of course, a number of species and a number of functional entities that exists in each one of these regions here. So, for instance, in the Central Pacific, there are 312 species of herbivores and 66 functional entities or functional groups, which are quite good rate, taking into account that there are only 87 functional groups in the world. So, once we have these for one region, of course, we can have for the whole world, and we can start to looking at differences and similarities. For instance, in the Central Pacific, Western Indian, and Central Indo-Pacific, the feeling, the volume, the functional volume of the herbivores is more or less the same. There's, of course, 1 or 2 things that are different, but there are regions with a high number of species and a high number of functional entities as well. As we go to other regions such as the Eastern Pacific, the Western Atlantic, and the Eastern Atlantic, the pattern is very different in the sense that we have you see, the feeling is not, the functional volume of the herbivores in one given region covers little of the total functional volume. It means that we have the number of functional entities is much lower in these regions here compared to the richer regions. Interestingly, in the Atlantic, although the number of herbivores in the Atlantic, than the Eastern Pacific, the number of functional entities here is higher than here. It means that we don't know exactly what happened in the Caribbean, in the Western Atlantic, that although we have more species here, they are more homogeneous in their functional structure. Okay, so that's just to show that this is a very interesting and a very it can be a little difficult to interpret sometimes, but it's a very powerful way of visualizing functional differences among regions or times of the year and so on. Okay, just to finish, what main conclusions here is that there is a huge variation in the structure of the herbivores assemblages according to the region, but according to the region. Okay, this functional structure as well is profoundly different. Especially, there is a marked difference between the Atlantic and the Indo-Pacific in general. There are important differences in the body size of these fish as well. We still don't know what drives these differences in body size. Maybe diet, maybe the way or the age of the family or so on. But these herbivores are very important for the reef functioning. So they are very a target group to study, to tackle some questions, and putting together this functional structure and with phylogenetic analysis would be probably of much interest in the future. If we know how the different feeding groups evolved and where they evolved and when they evolved, it will be of much interest to solve some of these questions. That's it. Thanks. Thanks. Any questions? Yes.
Maybe you talk about this. How do coral richness relates with
it's not richness, it's coral cover in some sense.
I know, but have you tried looking into richness? Is it directly...
Okay. I used those 5 factors only, but I had a list of 10 or 12 something. Coral richness, I think it was very correlated to coral cover, so I just removed it. But I tested. I tested other things as well. Yeah.
With the feeding related to the body size?
Yes.
The 2 last, I don't remember exactly the name, but the one that basically eat corals compared to the one that suck it, right?
The grazers and scrapers.
Did you look into jaw mechanics? Because I can imagine that to be able to eat corals or rocks actually, like the parrotfish do, you would need to have like a big jaw, and maybe you have more power having a big jaw than a small jaw.
Yeah.
And I can imagine that jaw and body size may be correlated.
Probably in some sense.
A lot of assumption. But
yeah, I'll tell you that this group here, the scraper, I don't know exactly the number, but almost all the species are parrotfish here. Yeah, there are 1 or 2 echanturids or surgeonfish, but most are the parrotfish. Yes, certainly the parrotfish, they have very specific adaptations for eating and so on, but I didn't look at these feeding mechanisms and so on. I mean, the feeding mechanism, what we are saying, the jaw structure and so on, is in some sense related to the fact that they are scrapers and that they are grazers. So all the grazers, they have a very a more delicate structure, not as powerful or as strong as the other one.
No, what I mean is, like, if you wouldn't make a little parrotfish, maybe it cannot eat corals because it's not strong enough.
Yeah.
If you would just make it a small parrotfish.
Yeah, but that's a problem because, and I'll tell you that this classification between browsers, grazers, and scrapers is very artificial. Okay, so there are lots of problems with that. At present, it's something that we can use more or less. We can classify the species and so on. But the parrotfish, certainly when they are small, I mean, parrotfish, they're not very, there's no small parrotfish, that's the thing. They're always, I mean, they're larger than 20 centimeters and so on, 20, 25. That's the maximum body size of the species. Okay. So when they are juveniles and so on, of course, they have different diets, and they're not scrapers, of course, but this size here is the maximum size for the species, not a measured size for food individuals. I answered your question.
I think the question here was, how do you remove the correlation from the factors that you use in the functionality classification?
Did I remove?
The correlation, the internal correlation of the factors that you use for classifying them in different functional...
For these three groups?
Well, all the groups that you made, I think they were...
You didn't use mouth size, do you?
No.
No, yeah, in this case.
No,
I
no,
think
but
he's just asking if
like
mouth,
these
jaw-
it might be other factors
Oh, yeah, sure.
that
Yeah.
might be correlated, and they are like skewing your
For the size.
multidimensional space.
Okay.
And how do you remove these
So yeah, so
in
the
correlation of the,
Yeah, I got
it.
among
The
the
traits, you mean?
among the
That's
traits.
a principle
so if
for
you-
net
if you
analysis.
These traits here. Yeah?
Yeah. So if you include, for example, the power of the joints,
Okay.
it would be
Correlated to the
one
size.
more information, or it would have the same information of the size, for example.
Yeah, no, probably. That's
think
why
that's it.
that's why
That's I think it's the question.
No, my point is, you go back to the previous slide, sorry. We make a mess here in the presentation. It's just like you say, yeah, I don't know why they are bigger and the other one is smaller, but maybe it's just mechanics. Basically, the parrotfish is big because if it's not big,
Yeah.
it cannot eat corals. Full stop.
yeah, it's possible.
Yeah, that's not. I'm not
And-
going
and-
that
and-
far in philosophy. I hope I
And
was.
it makes sense because these are the parrotfish.
Yeah.
These are the parrotfish. So,
But there's a lot of jaw mechanics done, so it's why maybe someone knows about that. So then you could say, hey, this cool guy
no,
with
yeah,
this
it's a
nature.
possibility. Yeah, it has to. I mean, it's the way it feeds, and there's some relationship with the structure of part.
I just threw a rock at you and then you deal with it.
Okay.
Thanks.
No.
Thanks a lot.
But okay, to answer your question, I don't know if the question or not. That's we tested the correlation between among the traits. That's why it's not a, most of the time it's not easy to find what traits we are measuring. So we are dealing with these traits. I mean, these traits have been using. People have been using these traits for some time, and we are not using traits that are correlated. So there's no I tested if there is some, but there's no very important correlation among these traits. That's it.
So one of the main conclusions is that geography, region of the world explains community structure. Do you think? The question is why? Is it because of differences in ecology among regions, or just differences in phylogenetic relatedness? It happens that Atlantic fishes are from different families from Pacific fishes.
I think it has to do with the evolutionary history of the region, and of course the phylogenetic history of the families, the assemblage.
Contingency, not
Yeah,
ecology.
I mean, of course, the ecology may play a role here. For instance, when I show the proportion of browsers, they're more related to temperature. Temperature was more important for them. But for the history in general, the phylogeny and the evolutionary history is more important. And that will be a very interesting thing to do, because we are putting that on temperate sites as well. And when we have this, because these are more tropical regions. If we put the temperate data, and I think that there are some regions, especially in the South, Australia and New Zealand, that's very important for the herbivores in terms of evolutionary novelty and so on.
So could you predict which region or which categories are vulnerable? So in terms of climate change, if you increase temperature,
Well, we can model this in some sense. We can, yeah, we can. I never did that, but it's something that it's possible to do. I mean, we can relate. We can start if we know that. The thing is that the temperature was not important in general, and when we talk about climate change, we are most of the time talking about changes in temperature. So yes. Yeah, and this is interesting if we say, remember that the plot that I showed with the different interactions and indirect interaction and so on. These are very interesting because if we change one of these parameters, we can see how everything changes or not. So, yeah, I don't know what may happen. There are a lot of people working on the effect of tropicalization of temperate areas at the moment, but I don't agree with most of the things that people are doing. They're doing some nice things, but the picture is much more complex than just saying, "Oh, I increase the temperature, so there's more fish here or less fish there."
Can I throw last rock?
Huh?
No, your percentage of different feeding types, are they based on presence absence or on abundances?
No, everything just presence absence.
And would that affect your analysis if they were based on abundance instead?
Yeah, it probably would change substantially. The things that it took 3 years to compilate this database with presence absence. So at present we have some data from, but it's very. We have some data from some places of absence of abundance of these fish. I'll probably be working on that this next year.
I just have in mind because in the New World, often there's like some species that are just by hundreds. Well, if you would go to Madagascar, there would be like smaller populations of different species.
Yeah, that's one thing that we know, that in the Atlantic and the Eastern Pacific, the abundance, the number of species per species is smaller. Yeah. Sorry, it's higher. Yeah. It's higher. Yeah. We have more species per each species.
Individual per species, you mean?
Yeah. While when we go to the Indo-Pacific, each species, the abundance of each species is lower. The richest, the more species there are. Matter of fact. So, but we don't have data for most of these sites. So at present we don't know. We're only dealing with the presence of the fish there. Yeah.
Another question. So does anybody has a guess why the true herbivores are doing so well in the cold areas or temperate areas?
No, but my guess is that the region, I mean, most of these true herbivores, they probably, we don't know yet, but probably the center of origin was somewhere around South Australia and New Zealand.
Oh, really?
So that's why probably they're more important there. But at the same time, they are more important in the Atlantic all the same. So we don't know what happened, what may have happened to that. But that's why I think that evolutionary history is very important in this. Yeah. Okay.